Hello students. Now before starting with unit 4, I'll just give you a brief introduction about biotechnology. Now as you can see in this slide that what is biotechnology. When we define biotechnology, it is the use of living systems and organisms to develop or make more useful products. ऐसी कोई भी टेक्नोलॉजी जिसमें लिविंग सिस्टम्स या ऑर्गेनिजम्स को यूज़ किया जाता है to make or modify products or other processes for specific use. यूरोपियन फेडरेशन ऑफ बायोटेक्नोलॉजी ने डिफाइन किया है इसे कि द integration of natural science and organisms, cells, parts thereof and molecular analogues for products and services. जब हम biological systems को use करके कोई products बनाते हैं, modify करते हैं, कोई process बनाते हैं या modify करते हैं for specific use, then we say it as a biotechnology. Simplest and the oldest form of biotechnology used till today is मेकिंग ऑफ कर्ड, दही जमाना एंड मेकिंग ऑफ ब्रेड. ब्रेड के अंदर हम यूज़ करते हैं ईस्ट और दही के अंदर एज यू ऑल नो दूध को जमाते वक्त जब है हम थोड़ा सा उसमें दही डालते हैं तो दैट इज़ कंटेनिंग लैक्टोबैसिलस बैक्टीरिया और उधर जो है दैट इज़ सैक्रोमाइसिस ईस्ट जो है. तो दिस इज़ द ओल्डेस्ट फॉर्म ऑफ बायोटेक्नोलॉजी. द टू इम्पॉर्टेंट technique which enabled development of modern biotechnology is the alteration of chemistry of DNA and RNA to introduce into a host organism to change the phenotype of the host. That is genetic engineering. कि आप DNA और RNA की chemistry बदल के किसी और host में introduce करो ताकि वो जो change आपने किया है that is expressed in the phenotype. And I hope you all know phenotype what is expressed and genotype the genetic makeup of an organism. And दूसरा है chemical engineering, maintenance of sterile ambience to enable growth of desired microbe eukaryotic cells in large quantities for manufacturing of biological products. Then genetic engineering, it isolates and introduces only one or set of desirable genes without introducing undesirable genes. फिर techniques used are creation of recombinant DNA, use of gene cloning or gene transfer to host. Recombinant DNA rDNA technology है. When inherited into the host DNA, ability to replicate due to origin of replication, that is it initiates replication. Alien DNA is linked with host DNA, it replicates and multiply itself along with the host DNA और इस process को हम कहते हैं cloning. Now these are all the basic things which you will be studying in the initial units. You can just go through these slides. You all know about vectors, plasmids, restriction enzymes which are known as the molecular scissors, etc. You kindly go through these slides and if you have any doubt you can ask. Now these are the 7 steps which are involved in the recombinant DNA technology. First, सबसे पहले हम आइसोलेट करते हैं जेनेटिक मटेरियल को. देन हम उस जेनेटिक मटेरियल को हमें काटना पड़ता है रेस्ट्रिक्शन एंजाइम से जो कि रेस्ट्रिक्शन एंडोन्यूक्लियसेस यूज़ किए जाते हैं. फिर उस फ्रेगमेंटेड डीएनए सेगमेंट को अलग करना, आइसोलेट करना. उसके बाद लाइगेशन मीन्स जॉइनिंग दैट डीएनए फ्रेगमेंट इनटू द वेक्टर में जाने के बाद उसे ट्रांसफ़र करना. नाउ इट्स एन rDNA, उसको होस्ट में ट्रांसफ़र करना. Culturing the host cells in a medium at large scale and extraction of the desired product. These are the steps. Now isolating genomic DNA from the donor से isolate किया जाता है. Fragmenting this DNA using molecular scissors, same processes जो हैं ये detail में explain किए हुए हैं. Screening the fragments for a desired gene, inserting the fragments with a desired gene in a cloning vector. Introducing the recombinant vector into a competent host cell. Competent means जिसमें वो capacity है कि जो उसको multiply कर सके और express कर सके. Culturing these cells to obtain multiple copies or clones of the desired gene DNA fragment. Using these copies to transform suitable host cells so as to express the desired gene. This is the entire mechanism of the process is shown. You can go through this process. Now gene cloning. Using bacteria to make multiple identical copies of a single stretch of DNA is gene cloning. It is useful in understanding eukaryotic genome. Then we have the cloning vector, any vehicle that inserts a fragment of foreign DNA into the genome of a host cell. जब होस्ट सेल में इंसर्ट करेगा डीएनए फ्रेगमेंट को, वो क्लोनिंग वेक्टर कहलाएगा. कि जैसे जेनेटिकली इंजीनियर्ड प्लाज्मिड, वायरस, दे आर यूज्ड इन gene therapy. Then these are the very basic terms. What is a gene? Gene is the unit of function, one gene, one character, recombination and mutation. Then the modern definition says that it is a unit of genetic information, unit of DNA that specifies one polypeptide. It includes coding as well as the non-coding regulatory sequences. Then you have the exons and the introns. Exons are the segments of gene that encode a mature mRNA for a specific polypeptide chain, and introns, जैसे नाम बता रहा है, introns in between. These are segments of genes that do not code for any mature mRNA. These are found in most genes in eukaryotes and in some genes of bacteriophage and archaea. Now the essential features of gene: they can duplicate themselves, they can synthesize a particular protein, they can determine the sequence of amino acid in the polypeptide chain. Now some terms, these are like recon, it is the smallest unit capable of undergoing recombination and crossing over. Muton, the smallest unit which can undergo mutation. Complon, it is the unit of complementation. Cistron, the portion of DNA specifying a single polypeptide chain is termed as cistron. Next are the essential features of the gene. It determines the physical as well as the physiological characters. Situated in the chromosomes, occupies a specific position which is known as the locus. They are arranged in a linear order. They occur in functional states called alleles. Some of them have more than 2 alleles known as multiple alleles. Some may undergo sudden and permanent change in expression called as mutant gene, which occurs due to mutation, may be transferred to its homologous, crossovers or non-homologous counterparts, that is translocation. Now gene cistron relationship: in prokaryotes, genes and cistrons are equivalent, and in eukaryotes, cistron is equivalent to the exon. That means prokaryotes are not having introns and eukaryotes have introns. Therefore cistron, जो कि एक यूनिट है जो एक पॉलीपेप्टाइड को बनाता है, वो हमारे अंदर या यूकैरियोट्स में इट इज़ इक्विवेलेंट टू द exons, and prokaryotes में genes and cistrons are equivalent. Now many a times you will come across words like cis and transposition. Cis position of the gene या transposition of the gene, जब जीन सेम क्रोमोसोम पे प्रेजेंट है एक होमोलोगस पेयर के, तो वो कहलाते हैं सिस पोजिशन में. जीन्स इन द सिस पोजिशन आर ऑन द सेम क्रोमोसोम ऑफ अ पेयर ऑफ होमोलोगस chromosomes. Transpositions, when genes are on different chromosomes of a pair of homologous chromosomes, they are known in transposition. Now this is genetic engineering. It is the ability to precisely manipulate DNA sequences from widely different organisms. The process requires ability to cut DNA, to insert foreign DNA segments and glue DNA segments together. It can be described, genetic engineering can be described as the introduction of manipulated genetic material into a cell in such a way as to replicate and be passed on to the progeny cell. Now gene manipulation is defined as the formation of a new combination of a heritable material by the insertion of nucleic acid molecules into bacterial plasmid or any other vector so as to allow their incorporation into the host organism, which are capable of containing propagation.